keyboard on this computer. All right. So we're looking at microbes, a little bit about their cellular nature, a little bit about their genomes. And then today we're going to look at, how do they get access to food? How do they deal with tough times? A little bit of physiology. And then the next topic will be a little bit deeper into physiology. And this will help you with your field guide process, because when you go to your field site and you're looking at the different habitats, you need to start thinking about, well, what could they be doing there? And what are they contributing to this ecosystem? And so that's part of what you should be highlighting in your field guide when you talk about the microbes. Here, we looked at this last time, just a really nice visual showing how important the cell membrane is to the microbes, and that they have their membranes chock full of all kinds of enzymes and proteins that are necessary to run a lot of different physiology. And not all of it's turned on all the time. They're constantly assessing their environment. They're trying to figure out what's around them, what kind of nutrients are there, and then they'll turn on the right genes to get things up and running and turned on. And then in the inside of this cell, you're seeing some basic, well, a lot of physiology. You have glycolysis, of course, which is going on most of the time for most microbes, just like it is with us. And then here they're talking about the citric acid cycle or the Krebs cycle. And but you see, of course, there's no mitochondria in there. So their way of generating ATP, that's stuck in the membrane. You can see various ATPases. Okay, so very, very busy, even though we can't see them with the naked eye, they're tiny, and yet they're chock full of all kinds of metabolic pathways. So when they start looking at the different microbes in their different habitats, and they ask the question, how complex are their genomes? And it really is influenced by their habitat. So what you're seeing here on the x-axis is genome size, working bigger toward the right. And then on the y-axis is the number or the frequency of those isolates showing up with those different genome sizes. The open boxes, the clear boxes, are free living. And gray boxes are the obligate parasites. No, sorry. Open boxes, free living. Gray boxes, obligate parasites. Black boxes, thermophiles. So obligate parasites are down here. The black are thermophiles. So you can see that depending on their habitat, they may have a very tiny genome size, closer to 500, all the way up to 5,500. Obligate parasites, that means they can't exist without their partner. And you can see they tend to cluster toward the smaller genome size because they give up some of their activities and let their host do it for them. So they can purge some of their genome. The horizontal lines, that's the kind of the patchwork here. Those are endosymbionts. Again, if you're an endosymbiotic organism, you have a very relatively cushy life because you're in a larger organism and you're well protected. And then because you're tied so closely to your host, you can purge quite a bit. Free living prokaryotes, that's pretty broad. That's really, really broad. But you can see that they kind of win in terms of the larger genome size. They need to have a lot of options available to them because they don't have a host to depend on. They're in, and this is again very broad. This can be all kinds of different habitats. So you can see that there's quite a range. Here you're looking at just one species. This is Burkholderia cepacia. It used to be called Pseudomonas cepacia, but they reclassified it into the genus Burkholderia. Anyways, here you're seeing habitat variability on the x-axis. So as you go out to the right, it gets more and more variable. Oh, I see they spelled that wrong. Forgot the A there. Habitat variability, and then genetic diversity on the y-axis. And you can see there's this nice positive correlation, right? It's going up with the greater variability, greater genetic diversity, greater variability, greater genetic diversity. And so when they drew the regression line, you can see that there's a nice positive correlation. So again, just trying to show that I didn't know bottomland was so diverse in terms of variability, but I guess it is. Here they're showing that when you have conditions that are highly variable, changing throughout the day, let alone, you know, the weeks and the seasons and all of that, you do have to have a larger genome, good genetic diversity in order to deal with that kind of habitat. So basically what that last slide was saying is that the genetic diversity is positively correlated with the environmental variability. And so again, we're looking at the selection pressure of the habitat that these microbes are existing in. And that is strongly influencing the size of their genome as well as what genes are turned on and off throughout the day. So all the time that you're thinking about these microbes, like for your field guide, you should be thinking about the different habitats. You know, if you go to a site and you happen to have a wood habitat, mixed deciduous forest, or maybe there's some grassland, maybe there's some water, maybe lots of rocks, soil, of course. So there's a lot of different habitats, and depending on how simple or complex they are, they will be influencing the microbes quite strongly. Okay. They've also looked at this idea, and you can, you know, in macroecology, they talk about these ideas too. You know, are they a generalist type organism or specialist? How, and this often is referring to their nutritional sources. And with microbes, you can see similar as what you think about with macroorganisms. If you're generalist, that just means that you have the capability to use a lot of different nutrients. And it's you're pretty good at that. You have similar efficiencies with all these different nutrients. Like, yeah, I can take that in and use it, and I'll take this one and use it, and it's about the same for me, you know? And so you have a lot of options. You're very flexible. Probably one of the better studied generalists is Pseudomonas putida. This is also a heavily studied microbe for its ability to do bioremediation. It's very good at cleaning up a lot of the toxins that we have put out into the environment over the years. And for Pseudomonas, they don't think of it, so to speak, as toxins. They just say, this is good nutrition for me. I'm going to take it in and break it down. Hopefully they break it down all the way to far less harmful substances that end up cleaning up the environment as they're feeding themselves. So they have a high versatility. That's why they were so attractive early on when they started hunting for good organisms to do bioremediation. Pseudomonas is one of the first that they hit upon because they're just so good at lots of different nutrients. You can imagine then that specialist means that you are limited to what kinds of resources you are capable of efficiently using. A good example might be something like Methylococcus, that's the genus name, and it feeds on methane, and that's pretty much all it does. And so it takes CH4 methane and uses that for its carbon source. And so we would say, yeah, low versatility. Okay, so whether you're a generalist or specialist, when you think about how microbes are obtaining their resources, it's certainly not like a gray wolf hunting down a deer. They have to think about, you know, they're tiny, tiny, tiny. So you have to think about the scale of them versus the nutrient they're going for. Some of the nutrients are huge for them, so they can't just take it in whole. So very often what you see happening is, well, with all microbes, what they do is they excrete enzymes out into their environment to break down those complex carbohydrates, for example. And then once they get broken down into small bits and pieces, then they're small enough to fit through their transport proteins, get them into the cell, and then they can start running their physiology off of that. So the fancy name for those enzymes are called exoenzymes. Just trying to emphasize the point that they're exiting out of the cell. So if you are a microbe hanging out in a habitat where there's lots of microbes, and you're all excreting these exoenzymes to break down nutrients, and then you absorb the breakdown products into your cell and start running your physiology, you can imagine that there might be some issues with that in terms of the idea that if you have a lot of microbes around you, I think you could imagine a situation perhaps where some of the microbes might be sitting there going, yeah, I'm kind of tired today. I don't think I'll excrete any enzymes. Let my neighbor do it. And then once they do their work, they break it down into all these lovely bits and pieces, take it in too. So I don't have to do all the work. I'll let my neighbor do it. Now I'm being very anthropomorphic how I'm describing that, but basically that's what they see happening. They see that the microbes can develop this laziness, some of them, and they actually gave it a term. They call it cheating, microbial cheating. And it's really fascinating to get into this literature. They actually call it social microbiology. I'm not kidding. They actually call it that. So just like how, humans can cheat and manipulate, they actually use those terms with the microbes and run some really cool studies in the lab to look at this idea of cheating and manipulation, deceptiveness. I mean, they actually use these terms when they're describing their data and thinking about what's going on in the microbial world. But you can imagine that this could be a thing, right? I mean, if you are a savvy microbe, you know that, it costs energy to make these enzymes, resources, energy. You have to expend some ATP most likely, and then to build these exoenzymes, all those amino acids, string them together, get a nice protein out of that, excrete it out of you. That's work and effort and resources. So you could see where some microbes might be going, I'm a little low on resources right now. I'll just let my neighbor do it, and that'll be good. Those enzymes get released out away from the cell, some distance. And so any nearby microbe that could be right there could be taking advantage of the breakdown products and scarfing them up before the cell that did all the work has easy access to those as well. And so they have looked at this pretty carefully, and the research can get pretty gnarly pretty quickly because they're often studying complex organisms and following their growth patterns in the lab and doing all kinds of statistics. But basically, it does kind of make you think like, okay, well, if there's a cheater out there getting all this kind of free food, what's to stop that population from pretty much like taking over? And the ones that are doing all the hard work, kind of phase out. And they've looked at these ideas. You would think that over time, the cheaters would win and become the dominant isolate in that population. But without going into too much detail, they're finding that doesn't really seem to happen. That there seems to be like checks and balances, at least when they're doing these experiments, where the cheaters end up kind of shooting themselves in the foot, so to speak, and they end up, their populations never dominate and persist in a dominant form. And so there seems to be ways that the other microbes kind of keep things in check from a variety of different angles, which we don't have time to get into. But so they tend not to win, the cheaters. They tend not to win over time. They will persist for a little bit, and then they'll start going back to working and producing their own enzymes. They've also looked at cooperation. Could there be this going on in these populations of microbes? So we know that microbes don't live in isolation. It's really hard to imagine any habitat where there's just one microbe hanging out all by itself forever. And so they are pretty confident in this statement that microbes in their natural habitat, they live in big communities, often very diverse communities. And what they're finding, in fact, this is becoming a new field of research in terms of synthetic biology, where they're looking at using microbes to produce useful products for human use and other uses. They're finding that they need to grow different microbes together and have them interact with each other, because they're finding that in order to either produce a product or break down a product, often you need many microbes to do this, or at least more than one. And so a lot of the studies right now, they're looking at pairing microbes, putting two microbes together and seeing what they do. That's often how they start. And now with better technology, they're able to get into like small consortiums of microbes that they're working with in the research on this. And so often we see it's a multi-step process where they're teaming up to break down products. A classic example is this one, where you have a group of microbes that are called nitrosifiers. These are just a couple of genera that fit into that group. You don't need to know that, but if you're curious. So these nitrosifiers, what they do is they can take ammonia and break that down into, this is nitrate, I'm sorry, nitrite. NO2 minus is nitrite. By the way, ammonia is toxic to pretty much all living organisms, including microbes. So the ability for them to work with it quickly enough, break it down and produce nitrite. And then, so what they're doing in this reaction, they're not really going for the nitrite, by the way. That's not what they're trying to do directly. This is just a game, so to speak, that they play in order to generate ATP. And I'll touch on that later. But basically what they're doing is in their physiology, they take ammonia and convert it to nitrite. They're getting their energy needs met. And then now they're nearby microbes like Nitrobacter or Nitrococcus. They go, oh, lovely. I like nitrite. That's what I use. And so oxidizing, they'll grab that nitrite and oxidize it and turn it to nitrate and spit that out. So you have to imagine the microbes, when I say oxidize, what I mean by that is they're stripping electrons off of ammonia in this case. So they strip electrons off of ammonia. They're using those electrons to ultimately generate ATP. Then as they're doing that, they have a waste product of nitrite. So they just excrete that out. Then a nearby Nitrobacter says, well, I'll take that. And they'll take that nitrite and they'll strip electrons off of that for their needs, for their ATP. And then in that process, they create nitrate as a waste and they, excrete that out. And so you can imagine if there's a bunch of different microbes living in the habitat, it's that idea again of one person's trash is another person's treasure. They're able to take those excreted products that are just waste to one microbe and they'll say, I can use that. That's awesome. I like that for my ATP needs to get that ATP. So this is like one of the first examples that they discovered of how microbes hanging out can do this process. And they're realizing this is super common. Microbes in all kinds of different habitats will do this. So basically, most microbes are doing this process of exoenzyme production, getting those out of their cell, breaking down those large products or polymers or carbohydrates or whatever it is they're going for. And then usually it's a consortium of microbes with different enzymes helping out in this process. So one microbe doesn't necessarily have to have every single enzyme to break down a certain complex carbohydrate, for example. It can have some of the enzymes, and if a neighbor has the other ones, then they can take it to completion and then bring in those products. And so they think that this is a pretty common strategy. Right now, what we think is a less common strategy is this idea where you have some microbes that are what we call predatory. And there's not as much known, not as many species known by far that can do this. It's pretty specialized, but there are some that we actually call them predatory because of the way they behave when they're going for their food and their nutrition. The classic example is Delovibrio. The B is silent. So Delovibrio is an interesting microbe. It is, well, let me show you, let me go. So Delovibrio is being shown here. So it's a bacteria, the green is Delovibrio. And what they're showing is a free swimming Delovibrio. It's swimming around, it's hunting. They actually use the word hunting. And then when it finds its prey, in this case, it's E. coli, most of the experiments they do is E. coli as the prey. What you see is Delovibrio will actually attach to E. coli and then get into the cell. We're trying to show that it's getting past the cell wall and kind of, it sticks in between the cell wall and the cell membrane and kind of shoves its way in there and hangs out in between the cell wall and the cell membrane. And then it starts essentially using all the nutrients E. coli has and starts using that for its own nutrition. So it's like a parasite, internal parasite. And then you can see they're trying to show that E. coli is dwindling. It's lost a lot of its resources. It's pretty much dead. And then once the Delovibrio has reproduced itself, it'll burst out of there, be free, and start this whole process all over again. So it's kind of like if you've ever learned about viruses, it's pretty similar to how viruses act. You know, they'll get into the cell, take it hostage, so to speak, make more of itself, use all the resources for its own gains, and then release out. So pretty analogous in that way, but this is actually a bacteria, not a virus that's doing this. And so they actually, you know, will use the word that it's hunting, it's got prey. And then you can actually, I've hunted down Delovibrio, so to speak. You can find it in, you know, at the wastewater treatment plant, the water that's coming in, the dirty water essentially, that's coming in. I've like sampled that carefully. And you do see a lot of Delovibrio here in Marquette. So you have to do a special agar plate assay. It's not that hard, but it's a specific experiment that you have to do to prove that Delovibrio is in there. And every time I've done that experiment over the years, it works. So there's Delovibrio usually in very rich environments like that, where there's a lot of nutrients. And so they'll be hanging out with, because there's a lot of microbes in that area too. So that's a good spot to hunt for them. This is another idea that was on that previous slide. The nematode trapping fungi as another kind of predatory microbe. So now we're into the fungal world, and they have some interesting mechanisms as well. Here is a nematode, a big thing. So a nematode, if you're not familiar, they're little tiny worms. And when I first learned about nematodes, I learned about all the bad ones, the ones that love to attack plants and cause disease. But since then, we've discovered, not me personally, but microbiologists have discovered that there's plenty of nematodes out there that actually are doing a lot of good things in the habitat, including helping plants. So there's some nematodes that harm plants, some that help. But basically, they love the soil environment, and so do fungi, of course. And so here, the thinner organism here, that's fungal hyphae that's growing, and you can see how this fungus is actually growing and producing what they refer to as lassos. So that's a hyphal strand that has grown into like a lasso. And what happens, you can see this one, this is also a lasso, and it caught a nematode. So what happens is the nematode is you know, moving its way through this area, and then it, poor thing, gets it wiggles, you know, through the lasso, and that physical touch of the nematode against the hyphae causes the hyphae to get an inrush of water in that area. And so the hyphal lasso kind of swells and constricts essentially down on the nematode. And then the nematode can't get free. And then the hyphae just starts excreting exoenzymes and starts breaking down the nematode. Kind of creepy, but they're there. They're everywhere in the soil. And some of these predatory fungi have the lassos. Some have like short little hyphal branches that just have a really sticky like knob at the end. And so if the nematode brushes against that, it gets stuck due to stickiness. So sometimes they'll use a sticky type idea instead of a lasso. But I mean, it's pretty, if you are a fungus and you happen to catch a nematode, that's a feast. You'll be full for a long time. That's a lot of nutrition right there. So the payoff is big once you capture something. So again, these are, they refer to them as predatory because it's like they are kind of hunting down their prey in a way. This is a little bit more passive than Delovibrio. So basically you have, some options here where microbes, depending on which one you're thinking of, they might have to just sit and wait. So a lot of bacteria don't have a way to easily move around in their habitat. So they're kind of stuck. They have to just sit where they are unless they can get moved passively by like an inrush of water, for example, like if you have a rain event into the soil, then the microbes can move with the rain percolating through the soil perhaps. But otherwise, if you don't have flagella or another way to move, you kind of just sit and wait. You can also attach to surfaces. So think about streams, especially with any kind of current, rivers and streams. Yeah, a lot of microbes don't want to be swept down to the mouth of the river. They want to stay put. And so what they do is have those appendages. So here you're seeing a stalked cell where they can attach to a rock or, a log submersed or to the bottom of the river perhaps. And then here they're showing that in this particular case, this is Caulobacter, you can have during cell reproduction, you can see how one of the progeny cells ends up being motile and can then move off to a new habitat. And hopefully, if times are tough, hopefully they can find something else, a new habitat. And then on the right picture, what you're seeing here is two rod-shaped bacteria with flagella. So these long structures are the flagella. And so those flagella are really quite good at helping the microbe get from point A to point B. But again, not all microbes have flagella. Flagella are very complex. I teach a little bit more, quite a bit of detail of it in Gen Micro. And in those of you who've had that training, flagella are complex. There's all kinds of bits and pieces to make it up. It takes a lot of ATP to get it to move, to actually get you to move your cell from point A to point B. So it's a definite investment in energy and resources. And so you better have a good reason to do it. And so I often find that the microbes I work with, I have lots of frozen microbes, stock microbes that I use for this class in Gen Micro. And I treat them very well. We grow them up in lovely, rich broth. They have everything they ever want. And then you can imagine in that scenario, some of these microbes that have flagella, they're like, man, I'm a lab rat. I'm getting everything I ever want. I don't think I want to invest in this. I don't need to move around to find my food. This person's giving it to me left and right. I'm golden. And I actually see this happen and it drives me nuts because in Gen Micro in particular, I have to teach about flagella. And so you want them to produce flagella and they won't. I had that one year where they weren't producing flagella. And so all of our tests came back negative when they should have been positive for movement. So now I've learned we got to kind of stress them before we give them to you. If we're trying to prove that they have flagella, to make them make their flagella. So bacteria have this ability to kind of, no, I don't think I want to make flagella. So it's not like it's something they have to do every time they reproduce themselves. They can opt out of it, which is pretty amazing that they're that flexible. Okay, another idea here, if you think about aquatic systems, so if you have a river or lake and you're a microbe hanging out, that's your habitat, you would probably want to be able to get to different depths, maybe at different times of the day, as you're looking for food, or maybe you're trying to escape predation. Microbes are food for other organisms, so they're getting preyed on too. Here you're looking at a microbe, it's a cyanobacteria, and all of these like yellowish things, those are the vesicles. And essentially, it's just these membrane compartments. Well, actually, they're more protein, and they are filled with air. It's kind of like if you do scuba diving, and you can pump up your vest with air to get you to a certain depth. So you can just kind of hang in that, 30 feet. I just want to stay at 30 feet and watch the world go by. then it's kind of that idea where they have the ability to regulate essentially how much air they have, so they can rise up closer to the surface. If you're a cyanobacteria, you'd want that, right? Because you're doing photosynthesis. That's one of their huge things. So rise up to the surface during the day, do all that lovely photosynthesis, and then at night you might want to come back down to avoid predation and get deeper into the water column and maybe hide away somewhere. And so gas vesicles is a pretty sweet strategy if you're aquatic. The image isn't in there?
Yeah.
Okay, I'll update the EduCat PowerPoint. But my alarm is going off, so we should probably shift. So there's a few extra topics I need to talk to you on the slide set. So we're going to continue on with this on Wednesday.